for an updated About Me video. So my name is Bree Bell. People know me by the bumbling biochemist, and as of July 2023, I'm a visiting professor of biochemistry at St. Mary's College of California. I'm trained in protein and RNA biochemistry, and I am on a mission to make learning, and especially learning biochemistry, fun and accessible for all. And so here's a little bit more about me and the bumbling biochemist. The whole bubbling biochemist thing actually started right here at St. Mary's when I was doing undergraduate research, and I wanted to explain what I was doing to my family and friends. I wanted to tell them about all the cool things I was finding in the lab and all the fun experiments I was doing. But when I tried to explain it to them, their eyes kind of glossed over and they kind of just nodded along. But I could tell that they weren't really engaging, which made me like, why? Because this was just so cool. And then I realized that they didn't have the background needed in order to appreciate how cool it was. They couldn't even really make sense of what I was saying because I was speaking in all this scientific jargon, all these technical terms that didn't make sense outside of this very specialized context. And they hadn't had the training needed in order to know what those words even meant. It wasn't that they were stupid and it wasn't that they weren't trying, they just hadn't been exposed to that language. It was like a totally foreign language to them. It was like Greek, and some of the letters actually, the things, abbreviations actually are Greek letters. But basically, they didn't have the background, they didn't have the education needed in order to appreciate what I was saying. But I knew that they had the knowledge, they had the smarts to understand the concepts. If I could just explain it to them in words that they knew, if I could use analogies to help them understand things. And so when I started doing this, then I was able to better connect with them. I found that I was able to also connect with fellow students and with all sorts of people. And I realized that being able to explain things like this wasn't just important to explain things to a more general audience, but it was also a really great way to explain things to fellow scientists. Because although as scientists, we tend to kind of try to use big fancy words and make ourselves sound all scientific, and sometimes these do have a specific role, these do have a purpose, but really, if we can just explain things in simpler terms, it makes it easier for all of us to connect to one another and understand things. And so I was finding that as an undergraduate, a lot of the resources I was finding were either very specialized in that scientific jargon, where you had to really know that not only science, but know that exact field and know all the terminology that's used in the field and know all the nuances and caveats and be able to read these big scientific papers that are really hard to get through. Or things were really watered down for a more general public. And I found that I wanted to do something kind of in between. I wanted to have resources that would be helpful to people that wanted that level of detail that you couldn't get from that general science, like pop science approach, those articles for a very general audience, something that would be useful for people like me, people like undergrads and grad students who were learning science, but maybe needed, they were still learning how to read those big papers. They were still learning how to do all this stuff. And there was just this big hurdle between them and that level of detail that they needed or that they wanted. Because sometimes people, even if you don't need that detail, the detail is just really cool because science is really cool. And so I wanted to provide that level of detail, but do it in a way that is understandable by a wider audience. And so this is what my mission has been as the bumbling biochemist. It's basically just a biochemist on a mission to make science, biochemistry fun and accessible for all. And by accessible, I really want that to be meaning in all senses of the word. So not only do I want to make it so that I'm talking about things in terms that don't use all that jargon, but I also want to make it so that there's tons and tons of free content out there so that people that aren't as lucky as me to have a tremendous education all throughout that I'll tell you more about, that they would also have access to this. And I know that I can't provide access in a lot of the ways that are most important, those of the resources, the actual like physical resources to access an education, to access grad school and lab equipment, because really, if you want to do research and you want to be a lab scientist, the best way to learn is really at the bench.
but I knew that I could help people make the most of their time at the bench. I could help people learn the concepts. And so this is really what I've been trying to do as a bumbling biochemist. And I've collected a lot of content, both on my blog as well as my YouTube channel. And this content includes all sorts of things about experimental methods, about classic experiments, and about the biochemistry fundamentals, the concepts behind all these techniques, that detail that you can't find in those more general descriptions, and the detail that's spread out and hidden in all of those scientific papers. And so I try to kind of weed through all that and boil it down for you. I talked a lot about jargon and how we use these technical terms, and I've been making it sound like it's this really bad thing. But really, there are uses for these words, especially when we're talking to scientists in the field, because these terms allow us to kind of distinguish between terms that are very similar and nuanced things. And so I think it's really important to be able to recognize those terms and to use them in the right context. But in you're speaking to someone who doesn't know that term, well, now you're putting up a barrier between you and them. And so what I do with my blog and with my content is I don't try to just avoid those terms. Instead, I introduce the terms, but I explain them in more general terms. And if I use them, I try to explain everything that I use at least once, and then so that people can follow along, and you'll be able to recognize that term if you see it and use it in the right contexts, but you'll also be able to kind of think about it in a way that makes more relevant, makes more sense to you. And so in addition to having explained these in my videos and posts and this sort of thing, I also have a glossary where you can find these terms explained. And if you view the blog posts on my blog, you can find that if you scroll over the terms, it'll give you a definition pop up if it's underlined. And then if you click on it, it'll actually take you to that glossary page. And a lot of these are complete with all sorts of infographic figures. And yes, I make my own figures unless it says otherwise. And I use Adobe Illustrator. A free alternative is Inkscape. And I have videos on how I use Adobe Illustrator and how I make graphics and that sort of thing, if you're interested. But I found that the graphics were another way in which I could really communicate things with people, because sometimes it's easier to just get an overview and get a really good understanding of things when you see it in this more visual layout form. So that's about me in the bumbling biochemist side of things. But what about me in the other side of things? In terms of my background, I was born and raised in California, and I attended St. Mary's College of California as an undergraduate. And yep, that's where I am now, back home. But it took me a while to get back here because I had to go off and do training. So after I studied biology at St. Mary's College of California, I went across the country for grad school to Cold Spring Harbor Laboratory. And after that, oops, that arrow's going the wrong direction, because after that, I went back from New York back to California, where I've done a postdoc at UCSF. And so a postdoc is basically where after you graduate from grad school, you go and you work in another lab to gain more experience, pick up new skills, that sort of thing. And so I've been working in Dr. Fujimori's lab at the University of California, San Francisco, and I'll tell you more about that in a minute too. And then after that, well, I just hopped over a little bit of across the bay, and I'm now in Moraga, where I'm a visiting professor of biochemistry at, you named it, St. Mary's College of California, my alma mater. And so let me tell you a little bit more about this. So St. Mary's College of California is a small liberal arts school. It's got about 2,000 undergrad students. We don't have, well, now they have like a master's of science program in biotechnology, but basically we don't really have grad students in the sciences. And so all of the research is undergraduate focused, and it is very small class sizes, and the professors get to work hands-on in the lab with students. So I'm going to be teaching biochemistry and biology, both for majors and non-majors. So it's a 1 semester for non-majors, like a survey type of course, and then a 2 semesters for biochemistry majors. And these are upper division classes, so I'm going to really be able to go in deep with them. Super duper excited. I'm also going to get to do some research on the side and hopefully do the summer research program with students next year. So I'm really excited about my role at St. Mary's, and I'm following in the footsteps of my mentor, Dr. Jeff Sigmund, whose lab I've actually kind of taken over as he's stepped into more administrative roles. He's letting me use his lab space. And I actually found a box in there in the freezer that's actually labeled from when I was doing research there. It says Bjorn Bell Sigmund. And so now I have extra boxes to add to it that are where I'm playing that leading role. And so it's really exciting. And I'm really grateful for Dr. Sigmund, both for letting me use his lab space as well as serving as a great mentor to me when I was at St. Mary's. So when I was there, I was studying this protein called neurolysin, which is this neuropeptidase, which basically means that it cuts up these little signaling molecules called neuropeptides. And I was studying how this protein worked by comparing it to other known proteins that more was known about, as well as trying to predict based on its structure how it would work. And so I did some cool studies on that. Nothing that reached the level of publication, but I've learned a lot, and it was a really great experience. I also, as an undergrad, went and did a summer research at experience at the Gladstone Institutes in UCSF with Dr. Steve Finkbeiner studying Huntington's disease, which is a neurodegenerative disorder that's a really, really horrific disease. And I was studying kind of the molecular basis for that through a grant from the Huntington's Disease Society of America. So at this point, when I was in undergrad, I was thinking I was going to be pre-med. I was pre-med. I was thinking I wanted to go to medical school. I knew I loved science. I knew I loved biology and biochemistry. I knew I wanted to help people. And the only way I was really exposed to being able to do that was by being a doctor. It seemed like the logical thing to do. And by doctor, I mean a physician, not one of the nerdy type of doctors like I actually ended up being. And so I was pre-med. I was all set to go to apply to med school. I even took the MCATs, studied for those MCATs and all that stuff. And then I realized that, well, I really, really love this research. And the more I did research, the more I, both at St. Mary's as well as at UCSF and the Gladstone Institutes, where I really got exposed to a more like research intensive environment of science, the more I realized I really, really loved science. And the more I realized that you can actually be a scientist as a career. And so this is what I decided what I wanted to do. And so I decided I wanted to apply to graduate school. Well, first I decided, okay, well, maybe I'll do an MD PhD because I've always been set on this medical doctor path because that's what I was exposed to. But when I realized that you could be a scientist, I'm like, oh, maybe I'll do both. And then the more I talked to people, the more it seemed to make sense to just go ahead and do that PhD because I didn't really see myself using that medical side of the degree. And I could still learn all that medical content and things like this of the medical context, and maybe even be able to help teach future doctors about the biochemistry behind what they were doing and help them better appreciate these things. And so I decided, okay, let's go to grad school. And so I decided to go from St. Mary's to Cold Spring Harbor Laboratory for graduate school. And this is a small research institution out on Long Island in New York. And there I joined the lab of Dr. Limor Joshua Tor. And the lab studies is a structural biology lab focus. So basically it studies how the shapes of molecules, their 3D shapes and structures, using techniques like X-ray crystallography and cryo-electron microscopy. And most of the lab was focused on really that structural side of things, and I did some of that. So you'll find I have a lot of content on X-ray crystallography in particular. But really, I was focused more on the biochemistry side of things. I liked figuring out how molecules worked and really connecting what they look like, those things that my colleagues were finding, into how they were working at the molecular level, these little like machines inside of our cells, which I just found so cool. So part of the lab focuses on DNA replication, so how your cells duplicate all their DNA before they divide. And then part of the lab focuses on these small RNAs and other RNAs that have these specific functions in the body. And so I was focusing on the RNA side of things, more in particular, this pathway, this RNA interference or RNAi, where basically these short little pieces of RNA are able to help regulate the levels of other RNAs in your cells with the help of this protein called arginine. And basically your cells make these little RNAs called microRNAs, and those bind to a protein called arginate. And then arginine uses those little microRNAs as kind of guides to go find sequences that have matching, that match. Well, I say match, they're really complementary, and I have much more on this in another post. But basically these sequences direct them to other sequences on messenger RNAs, which are the instructions for making proteins. And then the arginine goes to those sites and it shuts down the protein making the translation so that you get a decrease in that protein being produced. And in this way, your cells can regulate the making of various proteins. And I studied how this protein arginine was regulated through this process called phosphorylation. And if you want to learn more about that, I have a paper in eLife that I can direct you to. So that was grad school, and now I'm back at UC. So then I went back to California to UCSF. So the University of California, San Francisco, where I joined the lab of Dr. Denitsa Fujimori, where it's a chemical biology lab. So it kind of uses chemistry and biochemistry to really explore function in a biological context. So if you think about biochemistry, it's kind of like biology plus chemistry with a little more focus on the biology. When you think about chemical biology, it's a little more focused on the chemistry. So using chemicals and that sort of thing to get a better understanding as tools to kind of better understand and manipulate biological molecules. And so part of the lab focuses again on DNA side of things and part of it on RNA side of things. And once again, I'm focused more on the RNA side of things. So part of the lab focuses on like chromatin and methylation of chromatin, how the DNA is wound up and how various modifications to the proteins, the histone proteins that the DNA is wound up around, affect the making, the use of the genes. And so whether things are being proteins are being made and whether that sort of thing depends in part on the context of the DNA, whether it's wound up really tightly or loosely. And part of that is dependent on the modifications on those histone proteins. And so part of the lab is focused on trying to study the enzymes, so these molecules that basically help put on those marks and remove those marks from the histones. But that's not the part of the lab that I was working on. Instead, I was working on the ribosome side of things, on the RNA side of things. So I was studying the protein complexes that actually make proteins. And more particularly, I was studying antibiotics. So a lot of the antibiotics, the antibacterial antibiotics we use, including ones that we use in the medical field, as well as ones that we use in the lab, they actually work by inhibiting bacterial ribosomes. So they stop the ribosomes from making proteins. And so I was studying how these antibiotics affect those ribosomes. And this is still an ongoing work. And so hopefully in the future, I'll be able to tell you more about it, but I'm working to wrap up things on the project as I start my role at St. Mary's. And so I'm really, really excited about this role at St. Mary's, and it's going to really allow me to help more people and do this really in person with undergraduates. So I'm really excited about that. Going back to me and my philosophy, as well as like how I'm going to approach teaching. My basic philosophy is push electrons, not people. And basically, I try to avoid having these like insider type jokes. So I'll try to explain it really briefly. But basically, electrons are these parts of atoms. So in atoms, molecules are made up of atoms, and those atoms are made up of smaller parts. And some of those smaller parts are called electrons. And the atoms of the molecules interact with one another and form bonds with one another by sharing and electrons and that sort of thing. And so you can follow the path of these electrons in order to understand how these molecules will interact with one another. And much more on this in other posts. I just wanted to show that basically we have this nomenclature, we have this way of representation where we can actually track those electrons by using these like arrow pushing. And I love arrow pushing. I think it's so satisfying. And if you want to do more arrow pushing, take OChem. And more on this in other posts. But basically, we just have this, just know that there's this thing called arrow pushing. And I love arrow pushing, and I also don't like when people push other people. And I don't like when there's a bunch of competition. I don't like when people are mean to one another. I feel that science and humanity in general is helped out is best when we are being helpful to one another, when we're sharing knowledge, when we're doing all these things. So one of my least favorite things about science is all the competition and the secrecy in a lot of places. And I really think that the more that we can share, the better. And one of the things that I can share is my knowledge and my education, because I've been so fortunate to have such a great education and to gather all this knowledge. And so I really just want to share it with other people. And this is really the goal of my Bumbling Biochemist blog and YouTube channel and all of this content, is to really pay it forward and help other people learn as well. So I've been collecting, making and collating this content since those early days, where basically I started at the end of undergraduate, the summer after I graduated from St. Mary's. I started making this content. Originally, just I made these like little daily Instagram posts for my family and friends. And it was like a private account. And then one day, I don't remember exactly how it happened, but I took the account live and more people started to appreciate it. More people found it. And then I woke up one day and it was just kind of like my account had really blown up and there was tons of followers. And I thought it was some like hack and that there was it was all just some sort of like robot thing. But really then I started hearing from people and it kept growing, and there was all these people from students to actually professors and things like this that were telling me how helpful the content was. And I started to put this content together on a blog, and make things more formal, really try to increase my skills with that Adobe Illustrator. It takes a lot of time and practice, but I started to organize things on my blog, and really I've just been improving it over the years. And I'm going to try in these coming days and weeks to really organize it as clearly as possible. I've tried to do this by organizing things into various topics. So I have like lab techniques. I have that glossary I was telling you about. I have 365 days of science posts, pages on protein expression and purification, grad school thesis and postdoc related posts. I also have things organized into playlists on my YouTube channel, and I'll try to make it even more accessible. Because all of this, I have all this content and I'm not going to have all this time. And so I want to make sure that you can make the most of the content that I have. And so hopefully this content will be helpful, and hopefully it'll help more people really appreciate science. And if you're already in science, it'll help you better appreciate the things that you're doing. So if you're used to using kits, say, well, now I want you to be able to understand what's actually going on in those kits. And I want to help you be able to succeed with whatever path that you're, whatever resources that you have, to really make the most of them. And I don't care if you want to be a biochemist or you don't want to be a biochemist. I just want you to have the opportunity and the resources you need in order to appreciate just how really cool biochemistry is, and know that it's out there if you want it, and help you really understand it at a level that you can't find in easily explained terms elsewhere. So again, it's not about dumbing things down. It's just about explaining things in a language that makes sense, but not shying away from using those technical terms when needed, and making sure that I introduce you to those terms so that you can recognize them if they come up, and you can use them in an appropriate context. So that's me. And in the coming days and weeks, I will be starting to really do my course planning and all this stuff in order to start teaching biochemistry to actually like in-person students here at St. Mary's. And so I'm really excited about that. And I'm really grateful to St. Mary's for giving me this opportunity. And I'm grateful for Dr. Fujimori at UCSF and Dr. Joshua Tor at Cold Spring Harbor, and Dr. Sigmund at St. Mary's. And I'm grateful for all my colleagues here at St. Mary's. It's kind of crazy how they were once my professors and now they're actually my colleagues. And so it's pretty wild, and I still can't really believe it, and it doesn't feel real, but I'm really excited. And I hope that I can use this role to help inspire the next generation of biochemists, while also still being able to help people around the world through my online content. So that's me, bumbling biochemist, Brianna B. Bell. Yes, I'm still trying to figure out what I want my students to call me, because I am technically a doctor, but not like the medical kind or anything. So please don't ask me for medical advice. But I do have that PhD in biological sciences with the emphasis on biochemistry, focusing mainly on like protein and RNA biochemistry, and really just learning as much as I can about everything, though. So I hope this helped you understand who I am. I hope my content helps you understand biochemistry. And I hope that you have a really great day.